So up next, we have Richoo Davis, who is also doing some SNF related work, and we're excited to see his talk. He started his career in India, and from India moved briefly to Dubai, and then over to the States, where he settled into the freezing climate of Buffalo, New York, for his graduate research and his postdoctoral research. And he's now working with Prion-like and doing some really fascinating work on Prion-like domains and the functions of the SNF complex. So, Richoo, please take it away.
Just want to make sure my screen's good. Can you see my screen?
Screen looks good. We can hear you good. Yep.
All right, great. So yeah, thank you, Jackson, for that introduction, and also thankful to the Fragile Nucleosome organizers for giving me this opportunity to present our work to this community. So before I get into my work, I'd like to first acknowledge the people involved in this work. So this was a study that was initiated by Priya and Madhi did some bioinformatic analysis that led us to this in this direction. Taran was a then graduate student who performed some cool single molecule experiments, but I won't have time to talk about that today. And Anushka and Aishwarya also helped with experiments and their analysis. So our work has recently been accepted, but it's still in press, and so it's available on bioRxiv if anyone is interested to look at. So let me just get my laser pointer in. All right, so the FET family of proteins include first EWSR1 and TAF15. These are RNA binding proteins that are involved in many physiological functions such as transcription, DNA repair, and so forth. They're also associated with disease states, and they're often found as cytoplasmic inclusions in neurodegenerative diseases such as ALS and FTD, and also in FET fusion-driven oncogenesis that I'll talk about today. So if you look at the domain architecture, you can simplify them into two parts. You have a C-terminal RNA binding domain, and then you have an N-terminal Prion-like domain, which is a disordered domain that's enriched in few amino acids such as serines, glutamines, tyrosines, and glycines, and they're often depleted of charged and hydrophobic residues. And you can see that for all three proteins that these amino acids tend to compose a big portion of the domain. So FET family oncofusions are a class of pathological transcription factors. They are generated via genomic translocations. And this happens when heterologous chromosomes exchange paths, leading to in-frame gene fusions and subsequent formation of the fusion protein. In the case of FET oncofusions, the Prion-like domain gets fused to a transcription factor, and then you have the FET fusion protein that causes many pediatric cancers and leukemias. So here's a limited list of the FET oncofusions that have been discovered. And what you'll appreciate here is that the DNA binding domain is what determines the tumor type, whereas the N-terminal Prion-like domains are more or less interchangeable, which means that the Prion-like domains are driving a common mechanism for driving transcriptional programs that lead to cancer. Moreover, as I said, these FET oncofusions are pathognomonic, which means that the presence of the fusion is an indication of the cancer type. These cancers have low mutational burdens, and expression of the fusion in the right cell type is sufficient for the development of cancer. So what attributes do PLEs bring onto such transcription factors that make them so oncogenic? So lately, Prion-like domains have been shown to drive phase separation of associated proteins, including the FET family proteins. Phase separation is a thermodynamic process that occurs when homogeneous mixture separates into two distinct phases. So this happens as a result of interactions between PLE chains with each other. And on reaching a certain protein concentration called the saturation concentration, or CSAT, they demix into a protein dense phase and the protein dilute phase. So for PLDs, multiple studies from many labs have shown that the aromatics are drivers of this interaction. And as you can see here in the case of PUS-PLD, it has a distributed tyrosine network. And these are important because they can mediate a plethora of interactions, such as hydrophobic interactions, pi-pi stacking with self, as well as other aromatics and cation pi interactions. So, and which lead to phase separation. So using a canonical FET oncofusion for CD83, we wanted to know what happens when we fuse a prion-like domain to a transcription factor. So here are the parent proteins of the fusion protein. If you express, we do transient transfections in HEC293 T cells, and you can see that the first when you express first, it forms some small punctate-like structures, whereas the transcription factor DDID3 has more or less a diffuse distribution. But when we express the fusion protein, it formed phase-separated condensates. We can perform FRAP experiment to show that these are condensates that recover completely on bleaching, which means there is diffusion of molecules both within and outside the condensates, suggesting that these are dynamic condensates. So, and also other studies have shown that in myxoid liposarcoma cell line, where the protein is expressed, it can form condensates, these condensates which at their endogenous concentrations, which enrich transcriptional coactivators such as BRD4. So then how does FET fusion protein phase separation lead to oncogenesis? There was a seminal work by Miguel Rivera's lab working on the oncofusion EWS FLE1, and they found that the expression of this protein led to a change in the chromatin landscape at these GGAA microsatellite repeats. So it went from a closed chromatin state to an active enhancer. And for this, they were able to show that the chromatin remodeler MSY SNF was recruited to these regions by interactions with the EWS FLE1. So here's an IP of the BRG1, which is the catalytic subunit, and you can see that when they IP down BRG1 in these two sarcoma cell lines where the fusion was expressed, they could pull it down. So, however, if they synthetically brought the BAF complex, fuse the BAF complex to the FLE1 without any prion-like domain, that wasn't sufficient to to change the chromatin state. So this suggests there's an emergent behavior of prion-like sequences in driving these transcriptional programs, and that phase transitions may have a role in this program. So at the same time, Pierre-Amon's group also showed that the interactions with the MSY SNF complex is a general mechanism for FET oncofusions, and that this was mediated by the prion-like domain. So SNF, I don't really have to introduce it here, but as you can see in the structural studies, that this is a multi-subunit complex, and BRG1 or SMARCA4, while is sufficient for nucleosome repositioning in vitro, you need all other subunits for the functioning of the cells for their functioning in cells. So we first wanted to determine if BRG1 can interact with FUS-CD83 in the context of condensates. So what we did was to express BRG1 in HEC293 cells, and you can see that it's got a diffuse distribution. But then when you co-express it with the FUS-CD83, you can now see that FUS-CD83 condensates enrich BRG1 within that. So this is consistent with previous studies that show an interaction between FET oncofusions and BRG1. So we then wanted to ask what part of the FET oncofusions interacting with BRG1? Is it the transcription factor or the prion-like domain? Again, previous studies have shown that the FET N-terminal domain, which is the prion-like domain, is sufficient to pull down the SWI/SNF complex in cell culture lysates, suggesting that the prion-like domain may be sufficient. So in our test, this test if BRG1 can get enriched within post-PLE condensates, we wanted to test that. So, but FUS-PLE, when expressed in cells, they do not form condensates because of the high saturation concentrations. So we used a technique that was developed by the Brangwyn lab, where they fused a light-activated oligomerization domain to the FUS prion-like domain, and you have an mCherry tag for visualization. So on light activation, Cry2 oligomerizes, which increases the valency of interactions as well as nucleate phase separation. And when this construct is expressed in cells and you activate blue light, you can now see that these condensates being formed. So these condensates are dynamic, they fuse, they recover on trap, and if you turn off the blue light, eventually over a period of time, they will dissolve. So using this technique, we created droplets of FUS prion-like domain, and we asked if the full length BRG1 gets getting enriched. And as you can see here from the intensity profile, that these PLE droplets enrich the BRG1 protein. So then the question is, what drives BRG1's interactions with FUS-PLE, or if you want to make it a bit more broader, what drives MSY SNF's interactions with the FET PLEs? So what is missing from the structure studies and clearly evident from the few subunits that I've displayed here is that there is a significant disorder in M-SWI/SNF subunits. However, nothing was known about the role of these disorder regions on M-SWI/SNF function. Only very recently, a study showed that the N-terminal IDR of ARID1A was involved in condensation of the complex, as well as its interaction networks to drive chromatin remodeling activity at enhancer sites. So this implying that there is a functional role for its IDRs in targeting complexes to specific sites. So using the PLAC tool, we tool for sequence analysis, we showed that many of the ideas of M-SWI/SNF complex were Prion-like. And in fact, SWI/SNF complex was particularly enriched in such sequences compared to the other chromatin remodelers. So we decided to pick a few of these ideas that we thought were oncogenically relevant, as well as a catalytic, as well as BRG1, which is a catalytic subunit for further analysis. When we express these IDRs in cells, we observe that ARID1A, ARID1B, and SS18 Prion-like domains, they had a high propensity to form phase-separated condensates. And these are in the inserts, you can see cells at very low concentrations, which is measured from the mean fluorescence intensity within the nucleus, that at these low concentrations, there is no nuclear condensate formation. From in vitro reconstitution, we find that the saturation concentrations for these Prion-like domains are in the low micromolar concentrations compared to like the FET PLDs, which first PLD, which is at about 50 to 100-fold higher. However, PRG1 did not phase separate at any of these conditions, and that might be because of the low aromatic content that's present, as you can see from the sequence composition. So compared to the FAT PLDs, these PLDs tend to be much larger, and the sequences are a little more complex than what we see with the RNA PLDs from RNA binding proteins. So coming back to the question that we asked, what drives BRG1's interaction with the first PLD? We first tested if the folded domain could get enriched within this PLD droplets, and when we co-express this, we do not see any enrichment of the folded domain in these condensates, as you can see from the intensity profile. Further, when we then tested the BRG1 Prion-like domain, we saw that these PLD condensates can enrich PLD to some extent in these extent. And so which suggested that the PLD-PLD interactions can drive enrichment of proteins into PLD condensates. So we also further were able to test other M-SWI/SNF PLDs, ARID1A, ARID1B, and SS18, all of which readily form condensates. And when we co-express with FUS PLD, this is not the octo version, just the FUS PLD tagged to a M-cherry fluorophore. And you can see that FUS PLD enriches very strongly within these condensates. And moreover, from in vitro reconstitution experiments, we can see that these interactions are cooperative. So when we mix them at concentrations that were at concentrations that are below the respective phase separation thresholds, where they don't individually phase separate, but you mix them, they were able to co-condense into homogeneous mixtures, homogeneous condensates, as you can see from the images. So this images. And so then we wanted to ask if there is a specificity. I think this is an important question. Is there specificity in IDR or PLD-mediated interactions? So any functional compartment requires that selective partitioning be present, and that's also true for transcriptional condensates. And so work from Ben Chabri's lab had shown that IDRs can mediate selective interactions, and they partition proteins that are required for transcriptional initiation while keeping the negative regulators out. So when we look at the gene ontology characterization for PLD-containing proteins, we see that they are predominantly enriched in proteins that are associated with transcriptional activity. And so we reasoned that that a repressive protein such as FOXG1 with the contrary function should not interact with the PLD proteins. So a sequence analysis for FOXG1 showed that there was significant disorder in the N-terminal domain and with some prion propensity, and we use this region for further experimentation. When we created upper droplets of first PLD and co-express FOXG1, we now see that there is no enrichment of FOXG1 within this first PLD droplets, as you can see from the flat intensity profile. And this was also true in vitro reconstituted systems, that FOXG1 was actually being excluded from FUS PLD droplets. So these results suggest that PLDs exhibit specificity in interactions with functionally linked proteins. So then we delved a little into the question as to what sequence features allow for selective interactions between Prion-like domains. As I mentioned before, that the aromatics are key to interactions of PLDs, and that various PLDs are able to enrich within each other, which means there's some kind of a self-similarity there. And so we asked if tyrosine is also important for heterotypic PLD interactions. So here I'm showing you ARID1A Prion-like domain, and I've marked all the aromatics that are present. So, and what happens if, we can clearly see that FUS PLE gets enriched within the ARID1A PLE. So what happens if we remove away all the tyrosines? First thing we notice is that now ARID1A PLE has lost its ability to undergo condensation, which is consistent with the idea that these tyrosines are important for self interactions as well as phase separation. But however, when we co-express with the first PLD, after first PLD, we see that the after first PLD condensates can no longer enrich ARID1A, suggesting that they've lost interactions with the mutant. Similarly, we can also test this with a BRG1 Prion-like domain, which has low aromatic content, where we introduce 17 tyrosines to increase its aromaticity. While both of them do enrich within first PLD droplets, we can quantify the level of enrichment by measuring the mean GFP intensity within the after first PLD condensates. And you can see here that the mutant, which is aromatic, Aro plus, that what we call the Aro plus mutant, has a twofold higher enrichment within these condensates compared to the BRG1 Prion-like domain. So these results clearly suggest that tyrosine residues are key for co-condensation between FUS PLD and M-SNF PLDs, and we can tune the heterotypic interaction strengths of these PLDs by modulating the aromatic composition. So this is the last bit of experiments that I'm going to show you, and it comes to an important question, which is that are PLE-PLE interactions detectable in the absence of phase separation? So normally condensates are usually observed at very high concentrations and sometimes beyond physiological levels. So can we detect such interactions at concentrations that are physiologically relevant? So in order to answer this question, we performed a bead halo assay. So we use nickel NTA coated beads to attach hexahistidine tagged Prion-like domains to the surface. The concentration of PLD used is 250 nanomolar, which is at least 10-fold lower than the phase separation concentration of the Prion-like domains. And we also have an MBP tag fused to the Prion-like domain, and this just to make sure that this increases the solubility of protein of the Prion-like domain and prevents any surface condensation. So then we also attach a fluorophore for detection as well as quantification. And so for this experiment, we have the following setup. Here is the Oli beads control, which means there's nothing being coated on it. Then we have the MBP Oli control, which is just the MBP tag with the histidine, but has the fluorophore for quantification. We also have SS18 Prion-like domain fuse, PRG1 Prion-like domain, and the FOXG1 IDR. We then introduce the FUS Prion-like domain, which again at concentrations that are 400-fold lower than its phase separation threshold. The FUS PLE has a different fluorophore, electrofluor 594, and we see how well they get enriched within on the surface of these samples. So we know from our enrichment analysis within condensates that SS18 PLE has the strongest affinity for FUS PLE compared to BRG1 PLE, and I've shown you that FOXG1 IDR does not interact with the FUS Prion-like domain. So in order to quantify the interactions, we take the ratio of the client intensity, which is in the red channel, to the scaffold intensity in the green channel. And we were able to show across 3 trials here that on beads coated with FOXG1 IDR, the enrichment for FUS PLE is the same as the MBP Oli control or the background. And whereas SS18 PLE had the strongest enrichment and BRG1 PLE had an intermediate level. So these results suggest that interactions can occur at non-phase separating conditions, and also that PLE interactions are sequence specific. So again, we can also see a similar result when we in this experiment in cells with optodroplets, where we co-express optofus PLD with SS18 PLD. We are looking at cells where there is a very low concentration of SS18 PLD, where it's predominantly diffuse, there's not much phase separation. And what we see is that on activation of phase separation, we see that the FUS PLD and the SS18 PLD immediately enrich within the same condensates. So this implies that the recruitment of SS18 PLD to FUS PLD is not a two-step process, but the PLD existed, that these two PLDs existed as heterooligomers in the dilute phase and co-condensed. So with that, I'd like to end saying that we have a proposed model for PLE-mediated selective interactions that drive functional networking of transcription factors and activators that are essential for gene regulation, and these are mediated by disordered protein domains, but what determines the selectivity among the various the plethora of IDRs needs to be figured out. So I'll leave you with the title of the talk, which shows which says that the Prion-like domains can drive sequence-specific co-phase separation of chromatin remodeling complex with FET oncofusion proteins. Actually, I'd like to also acknowledge the members of the Banerjee Lab for this support and all the funding agencies. Thank you.
Again, that was great. Thank you so much. We'll jump right into another Q&A question. Who asked, could introduction of tyrosine residues in FOXP1 cause it to gain interaction in those droplets?
Yes, that's also one of our hypotheses, and something that we're testing is tyrosine residue sufficient for these interactions to happen?
And so with these, you take away the tyrosine in the ARID1 IDR or Prion-like domain, and you lose the interaction. Do you know what would happen if you replace the tyrosines with other aromatics? Like, is it specific to tyrosine, or could phenylalanine do the same thing?
Yeah, so there is work from other labs that show that all the aromatics are capable of driving these interactions in Prion-like domains. There's also a sequence of how strong these interactions are. So phenylalanine has the weakest interactions. Tyrosines have intermediate. In fact, tryptophans are much stronger in driving these interactions. So yes, you can replace them with other aromatics. So there's a lot of work from Mitag and Papu labs that have, and more recently from our lab too, that show these kind of behavior.
And so what then do you think drives there being sequence specificity? Is it the arrangement of those residues in the IDR, or what do you think?
At least in the case of these sequences that are Prion-like, we do suggest that tyrosines are the key determinants. Although we have conditions where we increase tyrosines a lot, but then they became like solid aggregates. So you need and they wouldn't interact with some other IDRs, which previously they could. So you need the patterning could matter in that case too. And so you need a good, I think there's an optimum number of tyrosines or aromatics, and that are important. Also, aromatics drive interactions with number of different amino acids. So it can also not just tyrosine, you can do tyrosine methionine, tyrosine proline. So you can also look at what the composition of the other IDR is. It may not necessarily have to have too many tyrosines, but if it has these other components, then it could probably interact with those too.
And then have you looked at all into phosphorylation of the tyrosines and whether that inhibits phase separation?
Yeah, I think there's already people who've done that. We are only looking into it, but there's work out there that shows that phosphorylation introduces more negative charges, and those are repulsive, and this could affect the phase separation of the protein. Yeah.
Great. Well, if there are no other questions, I think I'd like to thank both speakers again for 2 wonderful talks, and encourage everyone to keep an eye on the Fragile Nucleosome Discord, or join the email list, or follow the Twitter account. so that you're up to date with announcements and you know when the next talks will be, and you can look forward to seeing us again sometime in the future. So once again, thanks to Richoo and thanks, Katerina.
All right. Thank you.